Good morning, everybody. It's 8 o'clock, and welcome to virtual seminars in Precambrian geology. This week we have a presentation by Dr. Nick Butterfield from Cambridge University, talk titled "Co-evolution of Proterozoic Life and the Planet: Who's Driving What?" So we'll go for, as we usually do, we'll go for about 40 to 50 minutes, and then there will be a discussion session and question session. You can ask questions by typing them in the chat or raising your hand, and I'll do my best to get them in the right order. Next week, we have Roberta Rudnick from UC Santa Barbara, glacial diamictites and the evolving composition of the upper continental crust. So if you weren't in the meeting last week, I made an announcement to point everybody to our code of conduct page on our Google Sites page. So we do have a more official code of conduct now that whenever you enter a meeting, we're expecting that you've had a look at that and are ready to keep those points in mind and maintain everything we have outlined there. With that, I have no other announcements. So, Andre, would you please introduce our speaker today?
Yeah, it's a pleasure to introduce Nick Butterfield. Nick originally from Canada and did his undergraduate at the University of Alberta in Edmonton. And after it, he went to Harvard to do PhD with Andy Knoll and immediately started to work on microfossils from Greenland and Svalbard. And at that time was a large group of PhD students working on Neoproterozoic, including chemostratigraphy. And so that's probably rooted Nick's interest in co-evolution of life and environment. And at the same time, he started his interest on Burgess Shale and collaborated with several people from Geological Survey of Canada on study of microfossils from Arctic Canada. He moved after his PhD in 1992 to Cambridge to do postdoc, not surprisingly, on Burgess Shale, on fauna from Burgess Shale, and with Conway Morris. And I guess after 2 years, he came back to Canada, to University of Western Ontario, stayed for a couple of years and went back to Cambridge. And he works on a diversity of topics, mostly on microfossils in Proterozoic, but he also touched on Ediacaran fauna recently. He worked on Burgess Shale fauna, and his long-term interest in co-evolution of life and environment, which is what he will talk today. So I pass it to Nick.
Okay, thanks very much, both Andre and Alex. This is a great opportunity, great series of seminars. What I want to do today is indeed talk about co-evolution of life and environments. Who's driving what? I mean, the answer, of course, is yes. I'll just start off a pretty standard sort of diagram now. They're getting a little bit old, perhaps, but it's telling a really interesting sort of story that there's some biology going on, some evolutionary biology going on over the past 4.5 billion years. But that biology is taking place, on a rocky planet. So we need to understand what that rocky planet is doing. And there's an atmosphere that surrounds that rocky planet. And we need to know what's going on that front as well. So here's a great oxidation event seemingly going on at the beginning of the Proterozoic. At the end of the Proterozoic, there's a Neoproterozoic oxidation event. Both the beginning and the end of the Proterozoic seem to be particularly eventful with regards to environment, perhaps with tectonics, but perhaps with biology as well. We've got biology, curiously, starting out very early on, we've got a very early record of biology. It takes an awful long time for it to really get its act together and start seriously appearing in the fossil record. And as I say, the beginning and the end of the Proterozoic are particularly interesting in that regard. I just start off with this figure here. This is a standard Sepkoski diversity curve of families through the Phanerozoic. And there's no question that there's a lot of interesting evolution going on here. The comings and goings, radiations and extinctions of particular groups of organisms. And again, there's going to be an interplay of the physical environment and the biological environment. The thing about the Phanerozoic, of course, is there's lots of fossil record there to deal with. And you can really start to pick apart quite a bit of the story of what's going on, of what's influencing the other thing. What we're interested in, of course, is the record of life and its co-evolution in deeper time. So there's the Phanerozoic all squashed up into the end there. And what I want to do today is look at this Proterozoic interval, in particular, maybe the beginning and the end of it. Before I do, I just want to point out a couple of things that I think we should all know and appreciate about the world that we live in. It's quite a bit different than it's quite a bit different than the pre-Phanerozoic world. There's really superb overview paper by Baron et al. In PNAS a few years ago, looking at the distribution of biomass on the planet. There's just broad brush kind of calculations of where the biomass sits. And the punchline is this: the vast amount of standing biomass on this planet is found in plants, mostly trees, okay? And which is to say that standing biomass, a big chunk, major chunk of the biomass that we understand, it only appears in the Phanerozoic. That is something very, very different is going on as far as biomass is concerned in the pre-Phanerozoic world. Here's animals coming in. They don't really figure in too terribly much. And I think that feeds into a lot of the thinking that goes on as far as whether animals matter at all. Plants clearly matter. Here's a nice example of how we all understand that plants matter. Okay, embryophytes, vascular land plants come online in the Devonian, and in the Devonian going through into the Carboniferous, we see just catastrophic drop in atmospheric CO2 and an enormous rise in atmospheric oxygen. That's a direct consequence of a singular evolutionary innovation, namely the rise in radiation of land plants. There you have biology driving environment sort of in your face. There's no question about that. So just keep that in mind. As I say, animals don't really figure very much in the grand scheme of things as far as biomass is concerned. But let's just take a look at what's going on in the modern oceans. Okay, in the modern oceans, there's there's gazillions of organisms in the modern ocean, and most of them are microbial, right? So logarithmic scales here. Most marine organisms are small. Here's something to really appreciate when we're thinking about how the oceans work. This is a paper that just came out this year, again by the Baron group, doing the same sort of broad brush calculations. Really interesting. It's been thought to be the case for quite some time. They went out and actually looked around and determined indeed that was the case. But most marine biomass is constituted by large organisms, okay? By zooplankton, by fish, by larger vertebrates of one sort or another. Yes, there's gazillions of these small things, but there's an enormous amount of standing biomass represented by all of these larger things, which are animals, which are known exclusively from this interval of time back here. So all I'm getting at is that this world here is a fundamentally different kind of place as far as standing biomass is concerned, and all sorts of other things. Good. Okay. Let's launch back I want to just do a bit of a survey through the Proterozoic fossil record. And its pros and cons, its strengths and weaknesses, but stuff that we should all know. Going back to the early Proterozoic, this business of oxygen. Oxygen, as we understand it, as a lot of the storytelling that goes on has oxygen providing permissive environments which allow evolution to take place. And in that broad sense, we've got oxygen being the principal driver behind evolutionary innovation. We can look around in the fossil record. The first good convincing record of oxygenic photosynthesis of cyanobacteria comes from Hans Hofmann's group of the Belchers from the 70s. These are just absolutely fantastic, beautifully preserved microbial mats. And well, it's called Eoenophysalis belcherensis. To look closely at these things, all they really are spheroidal microfossils. They're fantastically well preserved. We recognize them as being cyanobacterial, not by virtue of their morphology, but by virtue of their behavior. They have characteristically cyanobacterial mucilaginous sheaths and so on. But fundamentally, these are pustulose cyanobacterial mats. And we can take this to the bank. We've got good, unambiguous cyanobacteria at 2 billion years ago. We can go back into the Transvaal supergroup. Here are some interesting filamentous microfossils that look all the world like cyanobacteria, filamentous sheaths of filamentous cyanobacteria. There's not a whole lot of morphology there. They could be like all sorts of other organisms could have the same sort of form. But again, it's sort of a sense in here that these things are behaving like cyanobacteria. You get a sense there's so horizontal versus maybe vertical versus horizontal, repeated horizontal microbial mat layers defined by filamentous microbes, to be sure. And it wouldn't surprise anyone if those were cyanobacteria. And we're talking 2.5 billion years, so just a few days before the GOE. Here's another thing that's worth appreciating, is that cyanobacteria are monophyletic. They're a one-off, they're an evolutionary singularity. And that's one of the things I'm going to keep on coming back to, is that some things evolve again and again through evolutionary history. And there are some things that really are just singularities. And cyanobacteria are one of those that only ever evolved once. And that's important. It's important to appreciate. And the question to ask that is, one of the questions to ask, of course, is to what degree they are driven by some sort of permissive environment, right? What is the cause of cyanobacteria? I'm not sure that we can look to permissive environments for a cause for cyanobacteria. The reason cyanobacteria are monophyletic is because they have this amazing capacity to oxidize water. Okay, and it's thermodynamic. You can read the quote. The oxidation of water is thermodynamically the most challenging multi-electron reaction in biology. It's almost impossible to do. It takes two reaction centers. It takes photosystem 1, photosystem 2. There's a water oxidizing complex in there. It's really, really tough nut to crack. And I don't think we can appeal to permissive environments for the evolution of cyanobacteria. I think it just is one of those stochastic things that appeared and as a consequence changed the rest of biological history, evolutionary history, Earth history, right? So that's point number one, is that we've got these cyanobacteria. I don't think there's a permissive environment kind of explanation for it. They just appeared and that changed evolutionary history. What about these guys here? What about eukaryotes? Eukaryotes are also monophyletic and they also turn up in the Proterozoic, okay? And then here's the last eukaryotic common ancestor. Here's 5 major sort of super groups of eukaryotes. And we won't worry too much about them. Here's the photosynthetic eukaryotes with primary plastids. Here's some amoebozoans. The episthecons include the animals and the fungi and so on. But the land plants emerge out of here. There is a last eukaryotic common ancestor down here. There's all sorts of extinct things that separate these constituent groups. But again, I just emphasize, eukaryotes are monophyletic. They're evolutionary one-off. And again, I think it's fairly straightforward to appreciate why that is. They're just super, super complex, a whole different kind of organism than those prokaryotic things. They've moved most of their information into the nucleus. They've got these mitochondria, these battery packs set about in the cytoplasm. That cytoplasm, very importantly, has a cytoskeleton made up of microfibrils. That's what all this green stuff is. And that allows eukaryotes to do all sorts of things that prokaryotes can't do. Okay. This is one of the most complex kinds of rearrangements of life you could possibly imagine. And it's a one-off, okay? You could say, well, those mitochondria, of course, have a capacity to carry out aerobic respiration. And so in some sense, there's a prerequisite of having oxygenic photosynthesis. But I think it's a bit of a push to suggest that oxygenation caused the evolution of eukaryotes. If it was as easy as that, there'd be multiple origins of eukaryotes. But in fact, there's only one. So what I want to do, just for a second, is poke around and take a look at the record of fossil eukaryotes in the Proterozoic. Here's a couple of examples. This one here appeared in the textbooks on a regular basis early on. It's a thing called Gripania. And it goes back to nearly 2 billion years or so. And back in 1992, it was described as a megascopic eukaryotic alga. Okay. And more recently, here are some macroscopic fossils that have been argued to be multicellular macroscopic eukaryotes as well. And that seems not unreasonable. Larger organisms typically are eukaryotes, but I think we need, in hindsight, we've made progress in this business. In hindsight, we need to tread much, much more carefully before we take these things to the bank and say these are eukaryotes, for example. And just for example, we now know that large size simply doesn't do it. There are large prokaryotic organisms that are more or less fall into this size category here. There are people who suggest, I think not unreasonably, that this could conceivably be, this Gripania organism, just a supergiant cyanobacterium. It's not entirely unreasonable. In a world without eukaryotes, there's potentially a niche for very large prokaryotes. There's advantages to large size. We'll come on to that. For these sorts of things here, again, I think we need to be very, very careful because there are colonial kinds of organisms that could easily mislead you into imagining that you've got frondose eukaryotic algae, when in fact, you might be looking at just a colonial cyanobacterium of some sort. So what do you need? How do you actually go about identifying unambiguous eukaryote? Well, remember that eukaryotes, the last eukaryotic common ancestor had that cytoskeleton. And the cytoskeleton allows you to do form, to do morphology, to make shapes other than with just simple turgor pressure. And that's what we're looking at here. There are a number of early Mesoproterozoic, late Paleoproterozoic fossils that fall into this category. These are unambiguous eukaryotic organisms given their rich morphology here. There's a variety of them, but perhaps the best known is Tapania. It's distributed worldwide. It's got all these little processes. It's got this funny sort of neck-like structure out here. Who knows what it is? But in order to build something like this, you have to be a eukaryotic organism. Here's another. So these Tapanias are late Paleoproterozoic, early to mid Mesoproterozoic. They're succeeded by a range of other ornamented spinos microfossils. And one of the best known is a thing called Tracheostrichosphaera. And here's a bunch of them, and they're known worldwide as well. And in a very crude way, you can use these biostratigraphically. Okay. Here's something interesting about these early spinos ornamented eukaryotes, is that yes, we can see them morphologically, but when we look around to the biomarker record, the correlative biomarker record, and we don't see the correlative biomarker. So in both the Roper group, 1500, the Lachanda end of the Mesoproterozoic, we're picking up Hoping signatures, but curiously, there are no steranes, and yet there's demonstrably, morphologically, eukaryotes in both of those units. And so what's explaining that? Where is that coming from? One possibility is that the capacity to biosynthesize sterols simply hasn't happened yet. This is a really interesting idea from Susanna Porter. And then much later in the day, you get that full suite of characters that defines extant eukaryotes. Another possibility is that we had sterol synthesis down here, but those eukaryotes were ecologically so minor they simply didn't figure into the biomarker signal. Both of those are up for grabs. My own hunch is that I think we've got full-blown crown group eukaryotes right back when we pick up Tapania in the first instance. Let me just touch on some things that are less ambiguous, that are guaranteed crown group eukaryotes. Bangiomorpha pubescens is one of them. It's now fairly well dated at 1,050 million years. Latest Mesoproterozoic, beautifully preserved. This from Somerset Island, and I won't go through the details. Suffice it to say that there is enough information here to identify this pretty unambiguously as a direct counterpart to modern red alga called Bangia. Okay, all the cell division patterns are pretty much dead on. The environment is dead on. It's a very, very good counterpart. I wouldn't say the same species, genus by any stretch, but we're talking about the same kind of organism back in the late Mesoproterozoic and living on exposed shorelines today. Just another specimen here. Liz Turner and I were up on Somerset. We actually pinpointed the locality. We can produce this stuff in volume now. Here's another. This is now a Neoproterozoic fossil called Proterozoic. It has less information, but it certainly isn't a prokaryote. It's got branching filaments. You can see the intercellular septa. It's got very large constituent cells. It's almost certainly multinucleate, and there's good modern analogs in this thing called Cladophora. And this takes us to crown group plants, crown group eukaryotes at 800 million. There's this thing here. This is just wonderful. This is just an incredible fossil. It comes out of North China, Tang et al. I'm really envious of these people. This is just beautiful, beautiful material. And it takes the record of this same genus by all appearances back to the latest Mesoproterozoic. Okay, you've got crown group eukaryotes at the end of the Mesoproterozoic. And here's something else, really just for fun. It's a really ornate thing, no questions asked eukaryote. using its cytoskeleton in extreme ways. It comes from the Shaler supergroup of Victoria Island. Again, it's early Neoproterozoic. Look closely at this. You can see that it looks like a single-celled creature, but in fact, it's got lots of intervening cross walls to it. This is a multicellular organism. And I argued for a while that it might even be a fungus. And the reason was that you can see that these things are growing out. This is the vegetative organism. It's growing out, you can see it's forming these loops. There's the cross walls again. But those hypha-like things are growing out and fusing. And that's what modern fungi do, and argue that, well, maybe we're looking at a Proterozoic fungus, and that'd be really interesting. I've lost my faith. I've become much more skeptical in my dotage. I think this is not good enough. I think there are ways and means of other organisms converging on this sort of habit. I don't entirely buy it. It's a possible fungus, but I can't get too excited about it. Just a couple of years ago, Lauren and co, again from the Shaler supergroup, published this really interesting beast here called Orosphera and described it as a fungus. And they added a character, namely they identified chitin, and it kind of looks like some modern fungi. I have to say, again, I'm a little bit skeptical. Chitin is certainly not known exclusively from the fungi. It's all over the protists, all over the eukaryotes. Probably might well be back in the last common ancestor. And again, I worry about the possibility of convergence on a form like this. At the end of the day, I want to know what I can actually take to the bank. I know this is for sure a eukaryote, and it could be a fungus. Moving into the Neoproterozoic, there's some new things. There's some new kids on the block. And this is Susanna Porter's work, but many others have been involved as well. We come into the late Tonian, there is a kind of a shift in tone about what's going on. And we pick up some new kinds of things, even as some of those others carry on. One of them is these vase-shaped microfossils, VSMs, and there's a compelling case for recognizing these as testate amoebae, okay? And there's the amoebozoans there, and there's some testate amoebae over in this super group here as well. But what we're looking at here are good crown group eukaryotes way up in these well-defined groups. We've got green algae and red algae we've already taken a look at there. Possible fungi over in the episcopods, okay? And then there's these weird things also turning up in the late Tonian. This is work that I did with Phoebe Cohen and Bill Schoff and others, and Andy Knoll's been involved. These are phosphatic scale microfossils. They're absolutely fantastic things and really unexpected. This is biomineralization, the oldest known biomineralization. It's turning up in the late Tonian, 800, 720 kind of thing. This was just a one-off for quite a while. These are fantastic things, and they're telling us that something is going on. So the record is very spotty, so it's hard to say, you know, whether this is the degree to which this is stratigraphically constrained. But I've got the wrong date here. Leanne Riedman and company just published, this is 2021, just published this study of some other phosphatic scale microfossils. Again, they're all falling into this sort of 800, this late Tonian sort of interval. There's something going on, it seems. Data are sparse. Something's going on in the late Tonian that's changing things, it seems. And it's in the late Tonian as well 800 or so, that we finally start getting a staring record coming online, okay? And I get a real sense that things are starting to happen in the late Tonian. And of course, right after the late Tonian, we hit the Cryogenian, and there's a climatic extreme, right? And somehow all of this stuff, is connected somehow or other. And how do we make any sense of it? Go back to this figure here. Here we are. This stuff is starting to happen here. And what's connected to what? What's driving what? Here's a paper, and I don't know if it's right or not. It doesn't matter if it's right. It's a great paper. Just, you can read the title. Just for thinking about things and thinking how things might be. Biologically induced initiation of Neoproterozoic snowball Earth events, okay? The argument here is that we've got this climatic perturbation, and we can think about it in terms of tectonics, of extreme weathering, of this, that, or the other. But what about if you change the nature of, say, the biological pump, which is what their story was, that could in and of itself change the nature of the atmosphere, and as a consequence, tip the system into a glacial world, okay? And that's the argument they made. By changing the nature of the biological pump, you can change the alkalinity of the oceans, draw down atmospheric CO2, and away you go. I think that's really, really interesting. That's a different kind of way of thinking about how things are connected one to the other, the correlations and the causal correlations. Now, they went on and thought about it a little bit more. So what might have changed the nature of the biological pump at this time? And what they noticed that, well, there's those VSMs, there's those scale microfossils. It looks like there's something going on as far as evolution is concerned. What might have changed the nature of the biological pump? And they suggested that something called eukaryotic, which is to say, maybe around about this time, eukaryotes started eating other eukaryotes. Up until that point, by implication, is that eukaryotes were feeding entirely on prokaryotic organisms. And this, because they have a cytoskeleton, because they can engage in things like major phagocytosis, that changes the game, and it changes the nature of the biological pump, and so on and so on. That's an interesting idea. It doesn't cut it as far as I'm concerned. I know that eukaryotes were engaged in phagocytosis, at least by the Mesoproterozoic, because we've got those photosynthetic eukaryotes. I don't think this is a big enough insult, to really change the whole carbon cycle. We're looking for something that's really going to kick the system in the pants here. So that introduces this thing. I don't know if Liz is here. This is really exciting stuff. Just in the last couple of months, and people sat up and looked and listened. I'm not sure if everybody bought it. Possible peripheran body fossils in the early Neoproterozoic. Suggestion is, from Liz, is that we've got sponge grade animals turning up in the early Neoproterozoic and carrying on, and potentially, I could see it potentially having something to do with that Tonian perturbation going on. They are very, very strange fabrics, and we can compare them with the fabrics we see in extant keratose sponges. And there's some Cambrian things that look kind of similar. It's worth worrying about. It's worth paying attention to. I know I'm inherently skeptical. And I talked to the press a bit and I expressed my skepticism. But it would make a really nice story, because I think, and I'll show you in a minute, I think these kinds of organisms can kick the system in the pants and really make things change. And that's mostly what I care about. I don't care what these things are so much as what they can do, okay? And how they can change the system, engineer novel environments. Anyways, one of the concerns about calling these 890 fossils sponges is that even the most generous molecular clocks, just pick that up. Most of the molecular clock, for whatever that's worth, not a whole lot, and suggesting that first animals, first sponges, maybe closer to 800. But let's not worry the details. Sponges are amazing creatures. They're very simple animals, but they're very complex organisms, and they have amazing function. Okay, lots of different cell types. They have 3 distinct tissue types. And I'm going to show you this video because you've probably seen me show it before, but it's just the most amazing thing you can imagine. Sponges don't have muscle, they don't have nerves, they don't have a lot of things. In some sense, all they are is just a bunch of fundamentally unicellular choanoflagellates stuck together. But they are stuck together, and they've got this apparatus, it's a pump casing they've built. And within that pump casing, they've got little pumps, and they're built of these collectivized flagellated cells called choanocytes. And they do a hell of a job. This guy is just introducing some fluorescein dye in and around these sponges. And those sponges, they just seem like dead logs kind of thing. I've never got very excited about sponges, except look what they do. It's amazing. Just the collective activity of all those flagella beating, just pumping water through those things, like it's going out of fashion, it's terracing through there, 20, 30 centimeters a second coming out the ends of those things. It's amazing what you can do, even with these very, very simple kinds of metazoans, but very complex kinds of organisms. And there's enormous amounts of water can go through. 1 square meter of sponge reef can process 350 cubic meters of water a day, which is to say, a column of water 250 meters deep overhead. It's amazing the amount of water that will go through there. At the same time, 90% of the picoplankton get filtered out, 12% of the DOC, and it just fundamentally changes what happens to that DOC. They're removing as much DOC in 30 minutes as free living bacteria do in 30 days. And Dan Mills has done some really interesting work with respect to the survivability of these things in low oxygen levels. I'll just point this out. The capacity for sponges to pump water changes all the rules of the game, okay? First of all, of course, Dan Mills is right, and how could he not be right? By that collectivized activity of all those flagellated choanocytes pumping that water through, that aerates tissues. If oxygen is a problem, if oxygen is the permissive environment you're waiting for before animals can evolve, it just doesn't work, right? If oxygen is a problem, getting together as a multicellular sponge-like creature, is exactly what you want to do. Look at this diffusive boundary layer. You start getting some flow coming through, and it retracts down to almost nothing, right? Flow makes breathing fundamentally easier. The other point I want to, so here's something I think is really worth emphasizing. We've got a single-celled, tiny single-celled aerobic organisms back at 1600 million, those first Tonians turning up on the scene. If they've got enough oxygen to carry on, there's easily enough oxygen for animals sponge-grade animals to evolve, okay? So it wasn't not enough oxygen. What was delaying the arrival of animals, of sponges, wasn't some sort of oxygen threshold, quite the opposite. The other thing I want to emphasize here is that that sponge pumping the pumping aerates sponge tissues. The pumping also filters and aerates the water. Okay, if you're interested in where aerated water comes from, well, I tell you one place it comes from is you put a bunch of filter apparatus in there and you'll get it. Okay, which brings me to redox proxies. I don't know anything about this. I'm not a geochemist, and I'll get shot down by the geochemists, but this is the stuff they're publishing, right? Here's uranium, molybdenum, chromium, iodine, calcium, redox proxies. And the overarching story is there's something fundamentally different once you move into the Neoproterozoic and into the Phanerozoic. Okay, we're talking sort of Tonian sort of territories as much as anything else. And that is a big part of the story for the Neoproterozoic oxidation event. And it's clearly a Neoproterozoic oxidation event. The question is it what's driving animal evolution, or is it a consequence of animal evolution? I just want to take so this is, the iodine calcium story is interesting. Just a few years ago, this is a Liu et al. Paper, looked more closely at the iodine calcium proxy. And really interesting story. Title is Late Inception of a Resiliently Oxygenated Upper Ocean. Okay. Iodine calcium, at any rate, tells us that the Paleozoic oceans weren't fundamentally different from Proterozoic oceans. They're all relatively low. We start getting a spike, an obvious spike in iodine calcium in the Devonian, and then it dies down again. And then we get this resiliently oxygenated upper ocean from the Mesozoic onwards. That's really interesting. Here, look at the explanation they offer. This is another figure from that paper. So here's the Devonian spike I'm turning up here. And they argue that this is driven by a fundamental rise in atmospheric oxygen. It's driven by atmospheric oxygen. They can't say that about this Mesozoic rise. And here they invoke a eukaryote offer a eukaryote-driven explanation, right? That is something to do with the rise in radiation, especially of these biomineralized plankton like coccolithophores, like planktic forams, turn up and radiate in the mid-Jurassic. And as a consequence of the evolution of these creatures, the depths of oxygen minimum zones increased, okay? There's no obvious shift in atmospheric oxygen, but rather there is a shift in the oxygen minimum zone because there's a difference in the phytoplankton, difference in the plankton, difference in the biological pump. So here's a much shallower oxygen minimum zone in the absence prior to the evolution of these things in the Mesozoic. They're getting it. They got the right idea, which is to say that evolutionary changes, innovations, have a capacity to really substantially change redox profiles in the ocean. Okay, and if I get time, probably not, I'll come back and revisit that. Okay, and just in case you're curious, almost certainly the rise in radiation of these kinds of plankton is a consequence of animal evolution. Anyways, I want to just take a look at world of shallow and deep oxygen minimum zones. Looking here at experimental lakes in northern Canada, now really wonderful decades-long experimental systems, sadly defunded and no longer with us. But what you're looking at here is a lake with a barrier across it. And on this side, it's all clear water and dominated by eukaryotic plankton. And over here, it's turbid water and stratified and anoxic at depth, and completely different story. And yet, the nutrients on both sides are exactly the same. The oxygen level on both sides is exactly the same. this is pretty standard procedure, of course. What we're talking about is very much the same conditions, but these two ecosystems are occupying two alternate stable states, okay? And there's a big hump between them, okay? But the point is this, is that the physical circumstances are exactly the same, but because they're occupied by different kinds of beasts with positive feedbacks on those particular environments, the whole system has changed, okay? Really interesting stuff. Snowball Earth is another kind of approach to this thing as well, right? You can be stuck on one side or stuck on the other. You have to get over the hump. How do you get over the hump? You know what the answer is. You get over the hump by introducing suspension feeding organisms and large suspension feeding organisms that can pump water, which is to say animals. Animals are exclusive in being able to do this because of their size and capacity to generate water currents. It's just a local example, small scale example, Rinkebein Fjord in Denmark. Whenever it was, 1996, this is just a horrible stinking cesspit. They opened this sluice here in 1996, let some marine water in that allowed the soft shell clams to get in, and everything changed. It went from being pea soup to being clear water. It went from being cyanobacteria dominated to eukaryote dominated, diatom dominated. Everything changed as a consequence of these guys here. Now, just moving ahead, well, there's a wonderful story, a similar kind of story to be told in the Great Lakes. The Great Lakes used to, the majority of them anyway, used to have that same problem. It was just filthy cesspits. And then there was in 1980 something or other, there was a Great Lakes Water Quality Agreement, and they quit pouring sewage into the lower Great Lakes, and it cleared things up a whole bunch. But and it tipped back and forth and so on and so on. Things really, and so here's the phosphate going down from the 70s, right? Okay, so trying to do something about the water quality in the lakes. Then the mussels got in, the zebra mussels and the quagga mussels invaded, and things changed absolutely dramatically, out of this world. Okay, so the zebra mussels come in here, and then the water. So this is water clarity. The water clarity goes way up, and that's due to these quagga mussels. And what's interesting, when those quagga mussels came in some sense, the whole seeming eutrophication business in the lower Great Lakes got worse. So there's really interesting studies, all sorts of concern about this. The Cladophora research in Lake Ontario, characteristics and implications for management. And washing up on the shores, these enormous mountains of this green alga, Cladophora. We've seen it already. It's a benthic photosynthetic green alga. And it's just fouling those lakes like it's nobody's business. And how does that work? And I just go back, the introduction of all of these zebra mussels, quagga mussels, those are crystal clear lakes any longer. There's nothing suspended in the water column anymore. It's a real concern. And yet, including nutrients, the nutrients have gone way down. And yet there's all this biological gunk piling up on the shores. Well, short story is, I'll just get this going. Here's two aquaria. There's mussels in one and no mussels in the other. Look at the history of quagga mussels in Lake Michigan. Nothing. And now literally the whole of Lake Michigan is carpeted in quagga mussels. Okay, what's happening? There's the phosphate has been mostly hoovered out of the system, and we're still getting all of this horrific Cladophora accumulation on the shores. the water has become crystal clear on account of what these guys are up to. There's a couple of things going on, and there's a really interesting study. This is from Hecke et al. Called the nearshore phosphorus shunt. There's a couple of things going on. One is because the water is becoming much clearer, these benthic photosynthetic algae can thrive at much greater depths than they used to be able to. The other thing is that the whole of the lake bottoms are carpeted in these things. They're hoovering out all of the nutrients. But the thing is that the ones on the margins of the lakes where the density is highest, they're capturing all these nutrients as a nearshore phosphorus shunt. They're capturing all those nutrients and then recycling them as pseudofeces, as feces, and so on and so on. So they're fertilizing what's going on the margins of the lake, even while the lake itself has become super eutrophic. There's almost nothing in the way of nutrients out in the middle of the lake. Has huge implications for the fisheries. And I think there's something really interesting, important to think about from that scenario when we're talking about the reorganization of marine phosphorus cycle in the Neoproterozoic. Okay, and here's Laxo and company suggesting one possibility, that it has to do with a bottom-up kind of process of sulfate reducing bacteria working because of the rise of sulfate, working at that phosphate and recycling it. I think there might be something interesting to look at with regards to this nearshore phosphorus shunt, insofar as this is also the interval where we're starting to get significant animal activity. Now, I see that I'm about to run out. I'll carry on for maybe just a couple minutes, Alex, if that's all right.
Yeah, go ahead and finish up, Nick. It's not a problem.
Okay. Here's Joachim Brox's story of, go back to the stearine business. Joachim Brox's story of the rise of algae in cryogenian oceans. And sure enough, there's really a substantial expansion of biomarkers. Once we hit 800 or so, there's the cryogenian, and you come out of the cryogenian and things are really expanding. How did Joachim argue for what's driving this? He didn't suggest it was oxygen, because after all, oxygen is a waste product of algae. Algae doesn't radiate in response to rising oxygen, quite the opposite. Invoked rising phosphate levels. Okay, though, I think we need to go back to this business here, right? These are exactly the same situation. What's different about this situation over here is got filter feeding metazoans in it, and this one doesn't. And there isn't a fundamental shift, as Laxo points out. There's not a fundamental shift in phosphate across the Precambrian boundary. It's really just where it's coming out, and how it's being partitioned, and it coincides remarkably, with the rise and radiation of animals. It seems to me. I won't dwell on this, except that we move into the Ediacaran, and we actually see bona fide large organisms which constitute significant biomass and can do serious kinds of things that microbes can't do. And these Ediacaran organisms, I know Greg will have different views on this, but I'm convinced that we're looking at metazoans here, stem group metazoans, or somewhere up here, stem sponges, stem cnidarians, something like that. The fact that they're large allows them to do things that small things simply aren't in the business. It's ruled out for small things. Few last pointers. Keeping in mind, going into the Ediacaran, we're starting to invent biomass. And that sequesters huge amounts of nutrients. You know, it allows those large organisms can do all sorts of things that small organisms can't. Biomass is one of them. Motility is a big deal as well. These are motile organisms, and all that stuff on the bottom is fine. But if you're really going to change the world, you've got to be up, moving around. And here is, I think this is a wonderful image. This is a depiction of food web in the North Atlantic. And mostly what you need to know is that there's primary productivity down here, and big fish eat little fish, eat smaller fish, eat zooplankton, eat phytoplankton. Fundamentally, that's how marine and aquatic ecosystems work. If you're interested in where organism size comes from, it comes from building up this food web. If you're interested where marine biomass comes from, it comes from moving up that food web. Okay, there's a lot of standing biomass and the associated concentrated nutrients in those big things. And those big things do a whole bunch of really interesting stuff. Because they're large and because they can move around, they can translocate stuff that wouldn't otherwise be translocated. Going up phytoplankton, zooplankton, going up that trophic structure here, and the scale of movement gets ever larger as you evolve and invent this kind of trophic structure. And this is just, you know, one of my current hobby horses. Something that I think everyone should know is that these things, these nekton, these things that swim around, you know, have an enormous potential to change how the marine carbon cycle works and everything else. One of the things they do is diurnal vertical migration. And migrating animals, powerful ecosystem engineers. They change physical environments by virtue of what they do. And diurnal vertical migration is just that. It's the most remarkable thing. Half of the marine biomass does it every day, twice a day kind of thing, dives down to depth. This is in the Red Sea, dives down to depth, 600 meters here, spends the day down at depth, goes up and feeds at night down at depth. What's it all about? Almost everyone's doing it. And it has to do with visual predation. All of those things in that food web are fundamentally visual predators, and everybody does it. Here's krill doing it, there's jellyfish doing it, eels doing it, tuna doing it, everybody does it. The things being preyed upon go down to avoid predators, and the predators all follow them down. Everything just goes down. Go down to depth, and at depth they spend the day down there, and all the stuff they've collected at the surface, they're down at depth, and they shit it out, and they die it out, and they metabolize it out, and they go back the next day and collect more of it. It's a really fundamental, important component of the biological pump. And I'm almost done. Oxygen minimum zones. Oxygen minimum zones are a big deal in the modern oceans, and there they all depicted there. 20% of the oceans are underlain by oxygen minimum zones of one sort or another. They're at different depths. Here's a really fascinating, important, interesting thing to know about oxygen minimum zones. This is a really fascinating paper. Intensification of open ocean oxygen depletion by vertically migrating animals. Here's what you need to know. The top of oxygen minimum zones coincides with the bottom of diurnal vertical migration. That's what is defining the top of that oxygen minimum zone, is the depth to which all those organisms are diving every day, and then leaving behind all of that biological oxygen demand. And for a metaphor, is rubbish removal, right? You cannot have nicely oxygenated surface waters unless you have regular rubbish removal. You can't have nice leafy suburbs without daily rubbish removal. And I think that's what's going on. What we're seeing with that neoproterozoic oxidation event, it is an oxidation event, but it's got nothing necessarily to do with atmospheric oxygen. It has to do with the changing structure of the ocean. So I can leave that here. What we're talking about, what I've been trying to convey is that who's driving what? Well, of course, there's a co-evolutionary thing. Of course, there are permissive environments, but there are also very, very importantly, biological contingencies. Some of these biological contingencies constitute very powerful ecosystem engineers that change things by virtue of what they do ecologically, metabolically. I think the test for bottom-up physical drivers is convergent evolution. If you cross the threshold and there's an opportunity and everybody jumps into it, then you've got a nice test for a bottom-up physical drivers. If, however, it's a singularity, there's only one thing doing it, it's not. It's hard to believe that it's a threshold crossing thing, okay? And my argument would be here is that a bunch of these things we're looking at, like cyanobacteria, eukaryotes, animals, and land plants, they're all evolutionary singularities. And we owe the nature of the planet we're living on, to their sort of accidental evolution. Sure, there's permissive environments, but it didn't happen automatically, right? So as I say, the Earth-Sun system, who's driving what? The Earth-Sun system is behind the wheel, but biology is changing the gears, he said. Thanks very much. I'll stop there. And what do I do? How do I get out of it?
All right, great, Nick. I mean, if you want to leave that up or move to whatever slide you think is going to please everybody, or just have it there for reference, that's fine. We're going to transition to some questions. They're coming in. People will raise their hand, send questions. And unless you need to leave, Nick, I'm just going to let it go. So let me know if you need to leave. But you have one from Gordon Love here at UCR. How do you sustain biological and chemical regional heterogeneities in the Ediacaran and Paleozoic and younger oceans if the marine system is responding to animal tipping point events?
Well, that's a good point. And let me put a qualifier in. I wouldn't suggest for a moment that the oceans are just very large lakes. There's fundamentally different dynamics, going on in the oceans. And so I'm not going to put any numbers on this. I'm not, this is as much as anything, it's an arm wave. But the fact of the matter is that when we move into the Phanerozoic, the whole system changes. We get this enormously greater amount of standing biomass, and that standing biomass does stuff. And it sequesters nutrients and so on. And a lot of those nutrients can be captured in nearshore settings and prevent them from getting out into the open ocean to a degree. And that can feed back on the engineering of those nearshore settings, right? The real point is simply this, that the Proterozoic oceans aren't just Phanerozoic oceans with a bunch of fish not swimming around in them. All of that stuff has a huge impact on what goes on. And the sponges and the cnidarians and all that coastal benthic stuff is controlling to an important degree what's going on in the whole ocean. I'll say it again, most of the biomass and concentrated nutrients are in those animals in the ocean.
All right.
I don't know if that usefully answers it. I'm not going to get drawn into arguing details. This is admittedly it's an arm wave.
Yeah, if Gordon wants to speak, he'll unmute himself. But I think that's good. Or Gordon, are you there?
Yeah, that's good. That's fine. Thanks.
All right. So Eric Sperling, your hand was raised next.
Yeah. Am I hopefully showing up? Nick, that was a great talk. I really enjoyed it, and I particularly like that you seem to be trending more towards a Tonian origin of animals, which I would agree with. And I would completely agree with you that the origin of animals is an evolutionary singularity, and I don't think oxygen had anything to do with the origin of animals. But when we look at the Cambrian radiation, I would see multiple lineages are increasing in body size. Multiple lineages are gaining armor, inventing new body plan innovations, et cetera, independently, which would make it a polyphyletic radiation. And so I was wondering how you would kind of compare and contrast the drivers for the Cambrian radiation with respect to the origin of animals and what you've been talking about here.
Well, that's exactly an argument I would make. I don't know, which is to say that polyphyletic radiation that you see with Cambrian explosion happening in multiple lineages, that is in your face. Convergent evolution, biomineralization, similar activities going on in different lineages and so on. That is based on a fundamentally new technology. That new technology is called an animal. You invent an animal, or you invent a bilaterian animal, and you open up a whole new universe of opportunities. But you've got that basic machinery, and all sorts of things are playing the game with that new technology. And I think that's all you need to know. But it took a hell of a long time to get that last common ancestor of animals. And until that point, nothing much was happening. Animals are incredibly insulting ecological entities, and they force the Red Queen to run. And there's predation, there's symbiosis, all sorts of things going on with them. But we've got a fundamentally new kind of technology, and there's this empty bucket to fill once you've got that technology. It wasn't there for 3.5 billion years. To put it a more simple way, what's the cause of the Cambrian explosion? The cause of the Cambrian explosion was the origin of animals. And it just took a lot of evolutionary tinkering to come up with that incredible new way of doing stuff. What's another interesting is that there's another incredibly complex thing that was invented. It's called a eukaryote. What's interesting is when eukaryotes got invented, nothing much changed. Eukaryotes have a lot of potential, a lot of latent potential, but until there's something really insulting, like a metazoan comes along and kicks it in the pants, and you're not going to see much except the boring billion. It's this categorically new, insulting kind of thing that gives us the Phanerozoic world that we're interested in. And then there's only one other categorically new, interesting kind of thing comes along, and that's land plants. That changes everything as well, of course. But it's a singularity. I like to ask this of strictly bottom-up people. What caused the origin of land plants? It sure as hell wasn't oxygen. There's no particularly good reason why the terrestrial environments through the Proterozoic shouldn't be covered with forests. It's just heaven for land plants. High CO2, low oxygen. There's no land plants there. Land plants haven't been invented yet. And they're an evolutionary singularity, a one-off. Once you get them, there's gazillions of land plants, not gazillions, there's 300,000 species of land plants now. But they go back to a single last common ancestor. It's inventing fundamentally new grades of organization, new technology. Invent the steam engine, and next week we're going to Mars.
Great, thank you very much, Nick.
All right, Susanna, your hand is raised. Then Porter.
Yeah, hi. Thanks, Nick. I loved your talk. It was excellent and fun to listen to.
Yes.
I had a question about, you said the test for top-down drivers is evolutionary singularity, and I agree with that. But I wonder how, when you're talking about really ancient events like the origin of cyanobacteria and, more generally oxygenic photosynthesis, the origin of eukaryotes, and of course, I mean, origin of any clade is a single event, but you mean something complex, a complex cell like what eukaryotes have. Given that extinction is so widespread, and we're talking about, events that happened over 1 billion years ago, 2.5 billion years ago, how do we really know whether there was an oxygenic photosynthesis evolving in some other clade, or a eukaryote-like morphology evolving in some other clade that is now extinct? And I wonder if some of those really early, say, macroscopic things like Ripania could be some kind of eukaryote-like thing that went extinct.
They could be. That's true, but you can contrast that, I suppose, with what we were just talking about with the Cambrian explosion, where there's lots and lots of different things that ended up playing the same game, and they're still with us, playing the same game. And there's no question. I know there's no question that oxygenic photosynthesis is demonstrably a really, really tough nut to crack. It's almost impossible. It's pretty close to magic, right? But you trip over it one day, and all the rules change. And I think that's true of those other singularities as well. There's the bottom-up permissive environment way of looking at things. Implicit in that is that evolution is easy. That you offer a permissive environment, and life will rush in and fill it all up, in fairly short order. And I don't. That's true for lots and lots of things. That's true for most, but not all, but most prokaryotic metabolisms. Seem to, when there's the opportunity, bingo, there's beasts in there, working away at it. Antibiotic resistant bacteria, COVID, all of this stuff, right? Bingo, these guys are in there as soon as there's an opportunity, filling in the holes. But that doesn't count for some of this other stuff. I got a colleague down the corridor, Conway Morris, who's written tomes on convergent evolution. I said, that's really interesting. Pervasive everywhere you look, except in the places where it really counts: oxygenic photosynthesis, eukaryotes, animals, plants, and so on and so on.
All right, I'm going to hop over to a couple of questions in the chat, and then we've got Shuhai. But Ross Anderson in the chat says, "Hi Nick, what do you make of documented onshore, offshore diversity gradients for eukaryotic microfossils, example in the Roper group, which have been attributed to nutrient limitations of early eukaryote ecosystems?"
That's a possibility. There's a number of possibilities. I wrote a paper years and years ago, noting that if you want to find interesting diversity in Proterozoic mudstones, you need to look at conspicuously shallow water ones. There's no point in going to deep water mudstones. They're all in the shallow water, interfingering with stromatolitic carbonates and so on. That's where all the activity is going. In a very simplistic sort of way, I could imagine it. Never mind nutrients. Just stratification. In my worldview, in the absence of marine animals, of suspension feeding animals, that oxygen minimum zone is going to be very, very shallow. It's going to look like that cyanobacterial pea soup to a degree, because there's nothing that's hoovering it out and moving it out of the system. So the oxygen minimum zone is very, very shallow. That Liu et al. Paper was getting the concept right for the Jurassic, right? I think it goes right back to the Metazoan business. Here's something worth knowing. I've had a bit of luck. Getting out fancy microfossils from Proterozoic mudstones. I have a search image. I'll tell you my trick. I look in shallow water settings. Those Tapania, lovely Tapania came out of, a few other things came out of the Belt supergroup with Zach Adam, work I did with Zach Adam a few years ago. That came out of the Grayson Shale, and the Grayson Shale overlies the Spokane Formation. The Spokane Formation is a red mudstone. You know, it's all really shallow water stuff. The Tapania comes out of the Grayson as well, right? But if you want to see where things are happening, it's in very shallow water settings. And why are they in very shallow water settings? Well, one, we're talking about benthic organisms. That's a whole other conversation. They're benthic organisms and they're aerobic organisms. And if you're going to be on the bottom and aerobic in a world where the oxygen minimum zone is only a couple of meters down, then you're going to necessarily be in very shallow water settings.
All right, we've got another one coming from Bob Stern. What do you think about the proposal that acceleration of evolution in the Neoproterozoic was caused by the transition from single lid to plate tectonics? And he cites a YouTube video there that I think I've seen before, but I think you know.
This is beyond my pay grade. I'm going to put all that in a box, you know, and hand it over for somebody to comment on that. Except that sort of explanation, you know, is as a driver is a bottom up permissive environment kind of explanation, and that's not the kinds of patterns I'm seeing. I'm seeing singularities here that change how stuff works. But also, don't get me wrong. You know, I know absolutely. I mean, tectonics matters. The atmosphere matters. Everything matters. It's just that, you know, certainly, I think in our sphere there is a proclivity to undertalk the biological, the evolutionary input, and explain things more commonly as bottom up, you know, physically induced phenomena.
All right, Shuhai, your hand was raised. Next.
Hey, Nick, can you guys hear me?
Yep.
Yeah, great. Thanks, Nick, for this great talk. I want to pick up from a comment that Eric Sterling made earlier, and that is, he commented that you seem to try on the Tonian origin of animals, and you know, that presumably has had an impact on the environment. For the top-down mechanism to work, it's not the origin of animals. One animal cannot do it. You need a lot of them to have an impact. So my question is, you know, for animals and for eukaryotes, how do you estimate the abundance of animals? And what if this, you know, there were no credible Tonian animal fossils, and in your model you need a lot of them. Yep. So you know, you can argue that, you know, they're not preserved or whatever, but the biomarkers for the eukaryotes, they really didn't. The eukaryotes biomarkers really don't show up in any significant amount until the Ediacaran time. There are some Tonian ones, but the ratio is very low. But exactly during the Ediacaran and the Cambrian time period, the change in Eric Sterling's 2015 paper argued that there isn't any significant change in the redox condition in the surface environment. There is change, but it's small change. No. So how do you put all this together? In, you know, the singularity is important. I think the key test is not singularity, but the abundance, right? Well, you have to quantify the abundance.
Well, touche at one level. I mean that's an excellent point. None of this, you know, first appearance doesn't matter. It's rising to you know, ecological domination, or at least, you know, so it can be felt ecologically. And molecular clocks tell different stories from abundance. Short answer is I'm not in a position. We're not in a position to talk about real abundance. We have to make a bunch of stuff up. Here's one of the things. First of all, there's a whole bunch of stuff going on in the late Tonian. Isotopic perturbations, climatic perturbations, and we're seeing bizarrely those phosphatic scale microfossils. Those are just bizarre. Those are just utterly unexpected. And what that tells me is, or it reminds me, just how incomplete the fossil record is. And if you're entirely soft-bodied, if you don't have a cuticle, you're just not going to register. And there's a gazillion amoebae around through the Proterozoic. Never mind through the Proterozoic. Today we don't see them unless they're testate amoebae. And the fossil record is seriously incomplete. What we can do is infer those paleontologically invisible things. That's what molecular clocks are trying to do, of course, as well. But we can infer paleontological invisible things by tracking the things that they're co-evolving with that aren't invisible. So those testate amoebae, the VSMs on the one hand, and then the phosphatic scale microfossils on the other, they may or may not be important in and of themselves, but I tell you what, they didn't go to all that trouble, so to speak, of inventing those robust skeletons for nothing. And it could be simple eukaryotic, but there is some impetus for that. And in my view, it's evolve or go extinct. And those guys evolved and did something in response to some really serious insult that appeared on the scene. And I don't see what the insult is, but I see the response to the insult. And I can't add it up and tell you how many soft-bodied invisible beasts. I agree. There is no evidence of Tonian, direct evidence of Tonian animals, but I might argue there's indirect evidence of them. In the shit is starting to happen biologically and also climatologically, of course. I can't do any better than that. I agree with you. The Sterain record, is that figure from Brock's paper looks really nice, but it's very, very spotty, and it could be most of that could be a preservational thing. Who's to say? I don't know. But one possibility, is that certainly the animals don't appear until in the middle of the Cryogenian or something. It's just my hunch.
Thanks, Nick.
All right, there's one next coming from Paul Hoffman saying, is tinkering time as a control on innovation a testable hypothesis?
Well, we know that evolution, I don't know if I think I know that evolution works by tinkering. And for really complex things, it's guaranteed to take a long time. And when you put the last piece of the puzzle in, and you end up with a new grade of organization, then things open up and new opportunities arise. Whether it's detestable, in some sense, it's just true. Evolution works by tinkering, but to test it specifically with respect to the record we're looking at, No, it's a story. And another story is, does oxygen drive the evolution of animals? No, it doesn't. Not even remotely does it. The fact that there's all this weird stuff happening more or less all around the same time beggars belief that none of it's connected to the other bits. Did glaciation cause the evolution of animals? I don't think so. Did oxygen cause the evolution of animals? I don't think so. Animals, the developmental business, the algorithm that goes into building even the simplest animal is one of the most complex algorithms that evolutionary biology has ever come up with. It's kind of comparable to that of oxygenic photosynthesis. It's a really, really tough nut to crack, and you can show archaean organisms all the oxygen you want. You're not going to get animals out of it. And it just takes enormous amounts of time.
All right, we have another question here from Jingjun Liu. Thanks, Nick. Great talk. A question: if the land was covered by cyanobacteria or algal mats as early as the Proterozoic, what innovative factor could drive the land plant evolution?
Well, too true. I don't think it's quite as challenging as in an aquatic system. You could have terrestrial systems covered with cyanobacteria, and you could imagine that being there could be a hysteresis there. You have to somehow displace the cyanobacteria in order to get those early land plants established. I can think about that. I haven't in the past. What I have thought about, is that the challenge of, after all, you go back a certain amount of time, the oceans, the primary productivity in the oceans at one stage was 100% cyanobacterial, 100% prokaryotic, and for the first approximation for some interval of time, 100% cyanobacteria. It's really, really tough to shift those cyanobacteria out of the system. The best way of clearing out lakes, is to introduce some suspension feeding organisms that are large enough to pump water and hoover out all that biological oxygen demand, because cyanobacteria thrive in that kind of world. Okay, and you're not going to get out of the cyanobacterial world by adding nutrients. For example, adding nutrients just makes things go more eutrophic. You need to get into the clear water situation. That's where where eukaryotic algae thrive, is in clear water systems. And the rise of algae, I might argue, is the rise of the environments in which eukaryotic algae thrive, namely clear water. You somehow have to get those cyanobacteria out of the way, and they're really, really tough. It's really tough to do that. Suspension feeding animals is the best way of doing that. And the other way of doing it is just to have super, super low nutrients. And we've got cyanobacterial oceans today, in the mid-Pacific gyres. You've got super, super low nutrients out there. That's not interesting. The question is, what does water look like where there is nutrients? On shelf seas and so on. What would they look like if it was just cyanobacteria? They'd be pea supers. And it's really tough, to get your foot in the door when those guys are so good at what they do. I don't know. I forgot what I was talking about. I'm just throwing it out.
All right, then maybe that's a signature that it's time to move to the next question. Malgrazada asks, Excellent review and interpretation. Thanks a lot. But what would happen in your evaluation if the green and red algae would be accepted as present since 1.7 GA and abundant and widespread and influencing the oxygenation of environments?
Yeah, so Stephen Bankston has argued that there are crown group red algae in the Paleoproterozoic, late Paleoproterozoic. I don't think in my worldview, it wouldn't make a huge difference. There's eukaryotes around early on, and there's some small, simple, multicellular, filamentous eukaryotes present at the end of the Mesoproterozoic. I don't think they're working away. They're finding a little toe hole here and there. I don't think that anything serious is going to happen until you bring in a really insulting ecological agent. And those are those things called metazoans in my books. And that changes everything, including standing biomass, diversity, everything. The Phanerozoic is defined by metazoans and metaphytes. And that's where all the diversity is. 10 million species, whatever, they're all animals.
I'm stopping the recording to keep the video at a manageable upload length.